So this video is about evidence of evolution, and this is the last video for test 5 and the last video for the semester. So hooray, you made it. So let's go ahead and get into this. So evolution is linked to geological events, and generally changes in the environment are going to select for changes in both body structures and molecules. And so over time, this produced the variety of organisms that we see today. So between the origin of life on Earth about 2.5 billion years ago and now, that's a lot of time to produce the variety of organisms that we see today. As it turns out, evidence of evolution is pretty easy to find. There are a number of different sources for this. Fossils, anatomy, molecular sequences, all of these get studied to learn how species are related to one another. And these get studied in context with the geologic timescale, which we went over in the last video. Now, this is something that we did at the beginning of this semester, and I thought that it merited being one of the last things that we kind of close on. So a penny is 0.061 inches thick, and if we stand pennies on end, how far do we have to travel stacking these pennies to meet common ancestors with other organisms? And so if you think about a penny roll, that's 50 pennies, and none of you are on your second penny roll, and I am, which is something that I'd strenuously object to, but most people don't reach the end of that second penny roll, and nobody has ever hit that third penny roll. So that's just 150 years. So how is it that we actually come to an understanding of just how massive and immense that time actually is? So let's take a look at some animals that are first more closely and then more distantly related from us. We're going to look at the animals, how long ago they diverged from us, and then if you were to lay down a penny for stand a penny on end for every year, how many miles of pennies, and yes, miles, would you actually have to travel? And we'll have some fun with this and see where you can get to. So starting at the Tazewell campus, if you start out with a chimpanzee, we diverged from chimpanzees about 4 million years ago, between 4 and 6, but I decided to use 4. So that's about 3.9 miles a penny. So imagine that you lay down pennies, you go down the hallway, somehow you manage to go down the steps, all the way out of the building, all the way down to the main road past Food City, then hang a left, you start heading towards LMU, and you can actually get to Bargain Barn Salvage about 4 miles down the road. And so imagine how long it would take you to stack all those pennies, and this is enough pennies to get you 4 billion pennies, okay? And so quite a distance already, and that's our most our common ancestor with our most closely related relative in the animal kingdom. And so the next set of animals that we're going to get into is dogs and cats. And so there is the best dog ever, Uga, clearly superior to other mascots, far superior to Smokey. I'm just going to go ahead and say that. I know you can't reply, but you're going to have to listen to it. Best dog ever. So we diverged from dogs and cats about 85 million years ago. Now, in pennies, you're laying down pennies for 81.8 miles. And where can you get to in 81.8 miles? Well, if you keep on going north, you can get past Pineville up I-75 to the Rock Castle River Trading Company, which I've never been to, but based on the pictures that I've seen, looks like a pretty cool place to go. Okay, so dogs and cats they're placental mammals, okay, meaning that they rear their young internally. They don't have a pouch, but the next thing we're going to look at is our split between us and marsupials, which are the pouched mammals. And the most famous example of that, of course, is kangaroos. Now, we diverged from marsupials about 140 million years ago. That equals about 134.8 miles. Now, if you go in a different direction, if you go out I-40, if you go head to Morristown on 25, get on the interstate, get on 81, get on I-40, you can go past Asheville to beautiful Swannanoa, North Carolina, which is a place I'd kind of like to. I'd like to visit Asheville in North Carolina because it just seems like really pretty country. Anyway, 134.8 miles in pennies. Imagine the kind of labor it would take to get you all the way past Asheville in just pennies and we're not even close to done yet. So another group of mammals is called the monotremes, and these are egg-laying mammals. They're some of the earliest mammals, and our divergence from them, including the platypus, is about 180 million years ago. And so that's going to translate into 173.3 miles of pennies. And where can you get to with this? You can get to Gordonsville. Now, I don't know why you would go to Gordonsville, but you can get there in pennies. So just for reference's sake, basically what we did to get to Gordonsville is we stayed on 81. We didn't take 40 to Asheville. We basically we stayed on 40 heading towards Knoxville. Okay, so get past Knoxville, get past Crossville, get past Cookville, and about halfway between Cookville and Lebanon, you have Gordonsville, and that's how far you could get laying pennies down edge to edge with 180 million of them. Okay, the next thing that we're going to take a look at is our divergence point from birds and reptiles. So birds are an offshoot of reptiles, so the divergence point between us and them would be essentially the same. And so there's a little reptile, and our divergence is 310 million years ago. Now that stacks up to about 298.5 miles worth of pennies. And how far can you get stacking pennies like this? If you go up I-81 in the different direction this time, go up past Roanoke, you can get to Lynchburg, Virginia. So Lynchburg is about halfway between, actually it's a little more than halfway between Bristol, the state line, and Richmond, Virginia. And as the crow flies, it's actually about halfway between Kingsport and Washington, D.C., so it's a pretty good ways up the road. So Lynchburg, Virginia, it's not Lynchburg, Tennessee, which is where the Jack Daniels distillery is. So I hate to disappoint some of you, but it's just the only city that I could find that was about 298.5 miles. Now the next divergence point that we're going to look at is from amphibians, and so there's a little frog there. And our divergence point from them is about 340 million years ago. That's the last time that we had a common ancestor with amphibians. This is going to translate to about 327.3 miles, and you can get to Columbus, Ohio in 327.3 miles. Now, I don't know why you want to go. I went there for an academic conference once, and they're all proud of being like the Ohio State University and all of that, but the city's a whole, basically. So, and if there are any people from Columbus, Ohio listening, yeah, I said it. Yeah, forget you and Ohio State. The SEC rules. There you go. Anyway, you can get all the way to Columbus, Ohio, laying down pennies with the divergence point from amphibians. Next on the way back, we've got bony fishes, and our divergence point from them is quite a bit earlier than that, 440 million years ago. Now, you can go 423.6 miles on this, and as it turns out, you can actually get all the way to Richmond, Virginia in pennies, again, laying down pennies. And if you'll recall, Richmond, Virginia isn't that far from Washington, D.C. So there's less of a distance between Richmond and Washington than there is between Knoxville and Charlotte, if that gives you any indication. So 423.6 miles of pennies. Our next divergence point is with sharks, the cartilaginous fishes, and our divergence from them is about 460 million years ago. You can get 442.9 miles, and perhaps very appropriately, where can you get to? Tybee Island, Georgia. Okay, so you can actually find shark's teeth on the Georgia coastline, so this one's really appropriate. But imagine this. I mean, think about this. So you leave Tazewell, you go out 25, you get to I-40, you take I-40, so it gets to I-26, you take that all the way down to Savannah. Then you turn east, and you got to go out to Tybee Island, Georgia. That's a long ways, and imagine laying pennies down one by one right next to each other that entire distance. This is how long ago that we diverged from cartilaginous fishes. Okay, so now we're going to take a look at other phyla. So we're going outside of phylum Chordata, and the most closely related other phylum to us is the echinoderms. So here's an example, a sea urchin. And so How long ago did we diverge from them? About 570 billion years ago. That equates to about 548.8 miles. Where can you get to? Mobile, Alabama. And so if you want a vacation on the Gulf Shores, you can get there and lay down pennies until you get there, and you'll have a divergence between us and the echinoderms. And the ironic thing about this is that you can find plenty of sea urchins on the floor of the Gulf Coast beaches. Next up is our divergence from arthropods. We diverged from them about 590 million years ago. That's going to be about 568 miles, and it's just about exactly 568 miles from Tazewell to the William Jefferson Clinton Presidential Library in Little Rock, Arkansas. So I really hope that y'all come to my library. You can lay down pennies and establish our divergence point from arthropods. But if you do that, just recall, I did not make you lay those pennies down. Okay, so next up is our divergence from, yes, I'm going to keep on going as if I didn't do that. Next is our divergence point from flatworms, the platyhelminths. And so there's a marine flatworm. Our divergence from them occurred about 630 million years ago. That translates into about 606.5 miles of pennies. Where can you get with this? To the University of Missouri campus in Columbia, Missouri. So you want to go somewhere and have an automatic SEC win? Go to the University of Missouri campus. We'll treat you nice there. They hadn't been SEC fans long enough to get nasty with us. Okay, next up on our list is jellyfish, phylum Cnidaria. Now, our divergence from Cnidarians occurred about 680 million years ago. That's going to translate into about 654.7 miles. Where can you get to with this? To Madison, Wisconsin. So maybe that kind of brings it home that you can get all the way to Madison, Wisconsin. Imagine laying down pennies just one by one little penny right beside each other, all the way up to Madison, Wisconsin. That's our divergence point from the Cnidarians. And we're not even out of our own kingdom. So there are more animals to be had here. Well, one more set. Continuing on, we've only got one more animal phylum that's more distantly related to us than the Cnidarians, and that's going to be the sponges, phylum Porifera. And so really primitive animals, not a lot of differentiated tissue. They're about as stripped down and basic as animals can get. So our divergence point from them is quite some time ago, even in geologic time, 800 billion years ago. Now that's about 770.2 miles, and you can get to Lawrence, Kansas like this. So great place for college basketball, not so much for college football. Plus, you know, Kansas is kind of crazy, so why would you want to go down there and lay down pennies? But anyway, if you did, then if you laid down pennies all the way to Lawrence, Kansas, that would be our divergence point from the Porifera. Now we're going to step outside our kingdom next and go to kingdom fungi, because we are more closely related to fungi than we are plants. So the next time that you see like a really slimy mushroom versus a really pretty flower, you're more related to the slimy fungi. But then again, I mean, who puts flowers on pizza like you do mushrooms? So anyway, some advantages to fungi. But our divergence point from fungi is about 900 million years ago. That's about 865.5 miles. Where can you get to with this? Boca Raton, Florida. You're almost all the way down past Florida. Yeah, talk about a crazy state to go into and through, but still, get down to way down south in Florida, and that's how far you can go on 865.5 miles of pennies. Next up, we've got our divergence point from plants, which happened about 1.5 billion years ago. Now that's going to translate into about 1,444.1 miles of pennies. Where can you get to with this? El Paso, Texas. So,
out in the West Texas town of El Paso, I fell in love with a Mexican girl.
sorry, couldn't resist that. But it's a long ways away. El Paso, Texas is way down on the Mexican border. So, you know, if you go all the way down there from Tazewell, laying down pennies the entire way, then you've got just enough space for the number of pennies in years Okay, now the final point of divergence that I'm going to cover is from bacteria. And so our divergence from bacteria happened about 2 billion years ago. Now that's going to translate into 1,925.5 miles. If you go on past El Paso, Texas, and get down into actually western Mexico, you can get to Guadalajara, which is a really beautiful vacation spot from what I understand down there. And so that's how far you can get on 1,925.5 miles of pennies. So I want you to imagine, again, you start laying down pennies in Tazewell at the Claiborne County campus, and you get out of there, and you go down to Highway 25, you go all the way to I-40, and you just start heading west, and keep on heading west through a variety of different interstates, and get across the border, and just, again, remember this, penny by penny, 1 penny per every year, not per day, but per year. And you'll get all the way down to the west coast of Mexico. So given that there's that many years between our divergence point, between us and bacteria, I think at that point it becomes understandable how you can have this fantastic variety and diversity of life. Evolution takes time, but we have had a long time with which to accomplish it on this planet. Okay, so what's the evidence for this? Lots of evidence. Paleontology is a study of fossil remains or other clues to past life, and fossils provided the original evidence for evolution. So we're going to take a look at fossils, some different types of fossils, and actually how they form. So even though these events that led to our diversity of life did occur in the past when no one was around to witness it, there are a lot of clues that suggest that all organisms have derived from a common ancestor. So this is going to be some fossil evidence, but there's a lot of molecular and genetic evidence to suggest the same thing. So as far as fossils go, it's evidence of an organism from more than 10,000 years ago. And these get really ancient. The oldest are over 3 billion years old. So this says that fossils document the history of all life on Earth. Well, really, no, they don't. It's a snapshot of some life on Earth. So not all species that lived left fossils behind, far from it. So fossils are a partial documentation of the history of all life on Earth. So fossils also allow researchers to test predictions about evolution. So basically, if you find fossils that are spread across in a certain number of years, then a lot of the time scientists are able to predict finding fossils with a certain set of characteristics, and indeed that has happened quite a few times. So there are a number of ways that fossils form, and we'll go over these. And the first two processes of fossil formation that we're going to cover are compression and petrification. So in compression, let's say that you have a leaf sink and nothing messes with the leaf. So it says, so leave me alone. It gets left alone, it sinks. Fine sediment covers the leaf, and the sediment will compress and form sedimentary rock, and what you'll get is a really nice leaf fossil out of that. Okay, petrification occurs when you have an animal, it dies, it decays, and is buried. Now, water that contains dissolved minerals starts seeping through the corpse of the animal or through the remains of the animal. And basically the organic matter is replaced by minerals and it turns to stone. This is something that is also called lithification. So petrifaction is lithification, turning of something to stone. So what you're actually seeing, say, with dinosaur fossils is you're not seeing the actual dinosaur bones. What you're seeing is sort of a stone model where organic matter has been replaced by minerals. Now, I don't want you to think that this means that you can't see a lot of detail in fossils. You find organic material down to the cellular level. You can find microfossils with minerals having turned to stone that can include the outline of things as small as nuclei. So you can have some pretty incredibly detailed fossils that get formed by this process. So here's an example of a petrified fossil. The next two ways of fossil formation that we're going to cover are impressions and casting. And so these are kind of opposite numbers of each other. So in an impression fossil, an animal dies and it makes an impression in the mud or the sediment beneath it. So a sort of a print of that animal remains. Now the animal decays away, but the impression that was originally made beneath it remains there. And at this point, the mud hardens to rock, and what you'll see is something that can actually carry some pretty fine surface details. So what you're seeing here is actually a fossil impression of scales. So this would be where an animal, scaled animal, died, and a scales pressed into the mud beneath it, and so you had this sort of impression fossil left behind it. Now, in a cast fossil, the animal dies and it sinks into sediment, and it'll get covered by sediment, and the animal decays away. However, the imprint fills with mud, and so instead of having an impression, you have sort of the opposite number of this. You have a cast of the original animal, because that mud, the imprint that's been filled, hardens to rock, and so you'll have really a three-dimensional fossil in which you have an imprint of that organism. And I've got a really nice cast fossil of a trilobite that I would have brought in to show you all. Okay, sometimes you get intact preservation, and this happens only very rarely because you have to be buried suddenly in the absence of oxygen. So what this is going to do is to minimize decomposition. It'll also prevent scavenging of the animal. So here's an example in which oozing sap traps an insect, and then the sap hardens into amber, and so you actually have the intact organism within the amber. So these are other examples of intact preservation. Some of these have been lithified, but the animals or the organisms are mostly intact. So, as I said before, even though fossil evidence is diverse, it's not a complete picture because organisms with soft bodies fail to fossilize, and fossilization is actually a fairly rare phenomenon. So, not all animals that die get fossilized. It's a pretty rare event, and so if you have a species that's pretty rare, perhaps none of the individuals actually get fossilized because there aren't enough of them. If you find a number of fossils of an organism, it means they were absolutely lousy on the ground, just bunches and bunches and bunches of them around, because if you get a lot of fossils, it means that there were a tremendous number of organisms, of which a few were under the right conditions to fossilize. Also, you get movement of earth plates destroying fossils. So we're going to take a look at how the tectonic plates of the Earth move a little later on in this video. And so sometimes the crust from one plate goes underneath another. Sometimes you get mountain formation and crumpling of the Earth. So you can get a lot of destruction of fossils from geologic activity. And also there's erosion. So even now, fossils might be eroding beneath you.
Eroding beneath me! Eroding! Eroding! Eroding!
Sorry, that was another one of those broad ideas that I had that I couldn't resist. Okay, so even though fossil evidence is incomplete, it does provide scientists with a lot of information. So these marine fossils are from Oklahoma, and that indicates that the area used to be under an ocean, which might be an improvement given that it's Oklahoma, but still it used to be under the ocean. And one of the neat things that you can see if you go to the overlook to the Veterans Overlook that's on Clinch Mountain, if you look at some of the rocks that they've got placed as sort of markers for parking, you can see I believe that they're imprints of crinoid fossils, which were marine fossils. So as it turns out, the Appalachians used to be a seabed that was later on built up in the mountains by tectonic plates colliding with one another. So we also used to be, even though we're mountains now, we used to be beach. And so you find crinoid fossils, and there are some pretty decent examples in the rocks that mark parking spots in that Veterans Overlook at Clinch Mountain. Okay, so how do you know the age of all these fossils? Fossils' age can be determined, and you use a number of different methods to determine the age of fossils. First of all, this just probably makes common sense. Most of the time, newer rock layers are on the top, older rock layers are on the bottom. Now, sometimes as a part of mountain formation, you can see these layers getting folded. So you have to be careful to follow the topography of what's going on so that you don't mistake rock layers that originally were on the bottom as newer. So this is something that's called relative dating. It assumes that lower rock layers have older fossils than newer layers. But as I said, you've got to be a little bit aware when you're thinking about this. It's indirect, it's simple, it's less precise, but it can provide some pretty valuable information. So a more direct method is to actually date the fossil itself. And one way to do this is radiometric dating. And this measures the amount of 14 carbon, which is a radioactive isotope in carbon, in a fossil to calculate how long ago the organism died. So take a look at the lower left of the diagram beneath the text here. So what you see here is a depiction of 14 carbon and 12 carbon. And so as long as an organism lives, it's going to take in some sort of carbon source in food. So the carbon that's found in the food, some of it is carbon 14. And so as long as we continue to take in food, we're going to take in 14 carbon, and 14 carbon is going to leave as nitrogen. So it does decay somewhat, but the original proportion of 14 to 12 stays the same so long as we're alive. Now, after death, because of course a dead animal isn't taking in any more food, there's no more new carbon, 12 carbon or 14 carbon added. And so this 14 carbon decays to 14 nitrogen and kind of disappears from the sample. So it's going to leave, it's still going to leave. So radioactive decay doesn't stop when an organism dies. It's a process that's completely independent of life. Now, of course, again, if you're not taking in new carbon 14, the proportion of 14 carbon declines. So if you take a look at what's going to be a graph on the right side, first of all, that axis is the percentage of the original amount of 14 carbon. So 14 carbon gets formed in the upper atmosphere in CO2. And of course, plants take in CO2 and they make sugars from it. And so it spreads all the way up through the food web. And so the percentage of 14 carbon in living organisms is about the same that it is in the atmosphere. So while you're alive, replacement of 14 carbon will equal the decay rate of 14 carbon. So the half-life of 14 carbon is about 5,730 years. And what a half-life is it's the amount of time that it takes for half of the carbon 14 to decay to 14 nitrogen. So your half-life of 14 carbon is 5,730 years. So if you take a look at this graph, it means that after death, half of the 14 carbon in the dead organism decays to nitrogen every 5,730 years, which means that the amount of 14 carbon decreases as time goes on. Now, think about this. What does it imply if you have 50% of the original amount of 14 carbon? That implies that 5,730 years have passed. So if you look at this curve, you can get a general estimate of how much time has passed based on the percentage of the original amount of 14 carbon. So look, if you find 25% of the amount of 14 carbon that you would find in a living organism, it means approximately 11,460 years have passed since that organism was alive. At 12.5%, approximately 17,190 years. Now, this only will handle fairly recent fossils. So let's move to the next slide and we'll talk through that a little bit more. So because the amount of 14 carbon decreases at a known rate, decays at a known rate, if you compare the proportion of 14 carbon to 12 carbon in fossils, including this woolly mammoth, you can calculate how long ago it died. And so you notice over the top I say it says recent fossils. It's because the half-life of carbon is a little over 5,000 years. So how would you measure a fossil that was 65 million years old? Well, you date other fossils using other elements. And so this is where you have to look at the geologic strata that surround it. So when you have a magma or lava come out to the surface of the Earth, its radioactive isotopes begin to decay because it's away from the radioactive inner layers of the Earth. Now, sedimentary rock is a mix of different rocks. It can't be reliably dated because it's got samples from a lot of different rocks that might have come from volcanoes or might not have. So sedimentary layers can't be reliably dated, but a volcanic layer can be. So the fossils that are in between the volcanic layers are considered to be between the ages of the volcanic layers above and below them. So if you look over at the right, you'll see that first top arrow says 495 million years ago, and the second arrow down says 510 billion years ago. And so if you find you can reliably date the volcanic layers, because the volcanic layers are undergoing decay of elements that have a much longer half-life. So if you find a fossil in between a layer that's dated 510 million years ago and a layer that's dated 495 million years ago, then this means that your fossil is between 495 to 510 million years old. And this is the reason why you see most fossils given an age range rather than a specific range. So again, here's a different picture of this. I wanted to show you just a second picture of this. And so you see the red layers. The fossil, the dinosaur fossil that you see would be between 60 and 200 million years because those are the closest volcanic layers that you've found to it. So the bottom fossil would be between 500 and 700 million years old. And so, yeah, there's a range, and 200 billion years is a pretty darn big range, but it's kind of the best that you can do if you don't have volcanic layers that are closer together. These are some of the elements that are used in radiometric dating. So you can see that there are some radically different half-life lengths in here. So there are a lot of half-lives that are a lot shorter than carbon-14, and you can see there are a lot that are longer than this. And so potassium-40 has a half-life of 1.3 billion years, and that means you can use it pretty practically in between 50,000 to 4.6 billion years ago. And so if you have a potassium-40 source in your fossil, it means that you can actually radiometrically date this. But it's much more likely that you find potassium-40, rubidium, thorium, uranium in the actual volcanic layers. And as I said, what you do is you find the volcanic layers, the ages of those, and then your fossils lie in age somewhere between the age of those two layers. Now, biogeography is something that's going to consider species' geographical locations, and as it turns out, the Earth's geography has changed drastically over the last 200 million years. So, all of the continents used to be joined into one supercontinent, Pangaea, about 280 to 200 million years ago. About 181 to 135 million years ago, you had the formation of two major continents, Laurasia and Gondwana. And so this is a result of the tectonic plates starting to move apart from one another. Between 100 and 65 million years ago, you see the present-day continents, they're beginning to form and beginning to drift apart. So eventually North America is going to separate from Eurasia. India is going to travel up and join into Asia. That's the reason why you've got the Himalayas in India. And because of the collision between India and Asia, and Australia is going to move a little bit further away from Antarctica here. And in the meantime, North and South America will eventually come together, and here you see today's continents, but they are going to continue to drift in their current directions. So if you were somehow able to go into suspended animation for about 50 billion years, when you woke back up, you really wouldn't recognize the map of the planet at all. Okay, and the reason why is because you've got large sections of crust called tectonic plates, and forces that are beneath them actually move the Earth's land masses. Continental movements, shape evolution, earthquakes, and volcanoes, these are evidence that Earth plates continue to move today. So earthquakes happen when one plate sort of slips along the other. And so I want you to think about this. If you start to push your finger along a surface, sometimes it'll sort of slip. It won't go smoothly. It'll catch and slip a little bit. And so as these continents move against each other, they'll catch and they'll slip, and the slips result in earthquakes. As continents push together, then you're going to form some weak spots in that joining between the continents. Between the plates, and you're going to find a lot of volcanic activity there. Okay, now the location of fossil species around 200 million years ago, this tells geographers that Earth's continents were arranged into one large landmass called Pangaea. And again, the reason why is because you see the range of different organisms across areas that are widely separated now. Think about how far separated that Africa and India and Antarctica are from each other now. They used to be joined together. And so Triassic land reptile called Lystrosaurus has been found in Africa, India, and Antarctica. Why would the same fossil be found in locations that are that far apart? The reason why is because those locations used to not be far apart. Now, there's a line in Indonesia called Wallace's Line, and this is kind of a demonstration that animals that are on either side of this line have been separated for millions of years and therefore evolving independently. So if you look at Java, Borneo, and Sulawesi and the Philippines, then you see clearly that some of the Indonesian archipelago shares fossils and animals that have more in common with Australia. Whereas on the left side of the line there, west of the line, you see animals that are more like the distribution of species in Africa. And so even though these animals are fairly close together nowadays, they used to be a lot further apart. And so east of the line, you see organisms that are much more like Australia. West of the line, much more like Asia. Now, anatomical relationships can reveal common descent. This is something that I think is really cool. We're going to look at something called forelimb homology. So what investigators will do is they'll look for anatomical features to determine the relationship of two organisms. And so this is a classic example of animals having similar bones in their forelimbs. And there's this pattern there of one upper arm bone and then two lower arm bones and then a series of smaller bones and then a series of digits. And so let's take a look at these bones one by one. And these are something that y'all are familiar with because we went over the skeletal system earlier this semester. So this is the humerus, it's the upper arm bone. What you don't find is you don't find animals that have got, say, 2 or 3 parallel upper arm bones. And so this is a very strong suggestion that they had a common ancestor that just had the one upper arm bone. So here's the ulna, and it's located in pretty much the same kind of orientation with all of these different vertebrates. Here's the radius, again located similarly. The metacarpals, which in us would be the wrist bones, and the phalanges, which would be the digits. Now, if you take a look at the bats, I think that one's pretty cool. Turns out that most of a bat wing is actually what would be our hand. So that's the framework for bats. If you look at the whale, then you'll see that the humerus is really short and stubby, and really stout, so is the ulna. And the reason why there is because there's a lot of pressure placed on that limb when an animal that big wants to turn or maneuver. So you've got to have really substantial and robust bones present to support the musculature necessary to change the direction of an animal that big. Some other things, if you look at the horse, then a horse has actually lost a lot of its phalanges. So it's only got basically one major phalanx left. So it's got basically one finger bone. So different selective pressures are going to drive the configuration of these bones in different directions. So, I mean, do you need to fly? Then you need to be light. Do you have the need for a relatively robust bone? Do you need to lose bones like the horse has? So, how these actually develop is dependent on the natural selection pressures that are being exerted on these organisms. Now, a vestigial structure is homologous to a functional structure in another species. So this has vestigial eyes in blind moles, hind limbs in some snake species, or at least the remnants of hind bones. Pelvises in whales, these are evidence of these organisms' ancestors. The one that I really like with us is we've got little muscles in our dermis called the erector pili muscles. And what these do is these cause goosebumps. So anytime that you get really scared or really cold, you'll get goosebumps. And this is a remnant of when our ancestors had fur. And so when an animal's fur stands on it, it's usually when it's scared or when it's cold. And if you think about an animal being scared and its fur bottling up, then it's trying to look bigger than what it is and convince you that, trying to eat it is not worth your time. If you've seen animals' fur kind of ruffle up when it gets cold, it's because if you increase the airspace between hairs, then that's going to have an insulating effect. And so it doesn't matter how hairy that a human being is, we don't have enough hair to pull that trick off. So for us, goosebumps are actually a vestigial structure because they don't really have a useful function with us. When our hair's down on end, they don't make us any warmer and they don't make us look any bigger. Now, anatomical structures are not homologous. If they're homologous, they come from a common developmental origin. If they are analogous, they look similar, but they don't have the same origin. So if you look at this raptor and this butterfly, their wings are used for flight, but the wing structure in birds and in insects is very different, and it has got different developmental origins. So analogous structures result from something that's called convergent evolution, and this is sort of a natural selection. This is a pressure that forces different organisms towards similar structures. So I think a real good example of this would be maybe fish and dolphins. There are a lot of people out there who think that dolphins are actually fish because they've got, the pectoral fins, they've got a fin on their back, and they've got a relatively similar shape, but whales aren't fish. And it's actually been selective pressures that forced that shape and the fins that have a useful purpose in terms of moving. Okay, now structures can reveal evolution. So if you look at this, in species 1 and 2, we're going to look at where wings arise first. And if species 1 and 2 have got wings, then it's usually going to be a shared origin because they're derived from a common ancestor. Now, if you look at species, in species 6, you had wings arise. But the wings in species 6 and the wings in species 2, these species separated from one another before either of their ancestors developed wings. And so they've got the same function, but their origins are independent of one another. Now, species 7 has vestigial wings that are homologous, the same developmental organism to species 6, but they no longer function. Okay, so as it turns out, embryonic development patterns give you evolutionary clues. So anatomical similarities, especially in vertebrates, are most often obvious in embryos. So take a look at how similar the human and the chimpanzee skulls are in fetuses. But as you move from fetus to infant, you start to see as as a result of growing to adult form, you're going to see these fetal bones deform, and they're going to deform in different ways. So if you look at the grid lines, you can see that you're developing different shapes. The jaw protrudes much more in a chimpanzee than it does a human, even as an infant. And if you look at the adult, you'll see where the brain pan of the chimpanzee has really flattened out, and the mouth is way out, the jaw is way out there, as compared to humans. Sometimes humans get called neotenous, more closely resembling their infant form. So you can see that the grid from fetus to adult is much less distorted in humans than it is in chimpanzees. And but your anatomical similarities are more obvious in the embryos than they are in the adults. So here's another picture of how early vertebrate embryos can look alike. If you look at the fish, the salamander, tortoise, um, the chick, and the human, you see things that look pretty similar. But as you go further into development, then they're going to start looking very different. And of course, by the time that in late development, then they're going to look, of course, very different. The evolutionary relationships between these organisms, humans, chicks, tortoises, salamanders, and fish, are much more apparent in the embryos. So earlier on, you're going to see much more similarities. But there's a saying in evolutionary biology that ontogeny recapitulates phylogeny, meaning that form follows evolution. So there are types of genes called homeotic genes. These are necessary for body patterning. What they're going to do is to control embryonic development by controlling the cells around them. So if you get small differences in gene expression, so that basically means when does the gene turn on? When does it turn off? Is it present in the same locations? Is it present in the same locations but different times? Is it present in different locations at the same time? Is it present in different locations at different times? So in this example, you have two genes, A and B, and they're responsible for patterning the forelimbs and the hind limbs. And when you have this particular location of genes A and B, then you get this particular form of development, or this particular developmental pattern. Contrastingly, when you take a look at a python, the forelimbs are going to be completely absent. The hind limbs are going to remain vestigial. And you can see that you have these genes expressed in different locations. And to be honest with you, I've read it when I was in grad school. I looked at a few articles on snake evolution, and so sometimes the genes are entirely absent, and sometimes they're expressed over a much shorter period of time or in a much smaller location. And so this is how you get a difference between a limbed and a limbless organism. When you start messing with genes that cover body patterning, you can get really major changes in body pattern from relatively small mutations. Now, this is a mutation in which a gene that would normally say legs need to develop here or antennae need to develop here. So the gene for antennae development in this mutation has actually been mutated to a gene that calls for leg development. And so where this fruit fly would normally have had antennae, now it's got extra legs. Okay, there is yet more, because sometimes gene expression is under the control of other genes, particularly called enhancers and promoters. So take a look at the diagram on the left. This is a gene called yellow. And again, don't know why they did this, but yellow doesn't mean yellow color. In this case, the gene yellow means black color. So if you look at this in the wings in the ancestor, the yellow expression, there's none of that gene expressed in wings, so there's no coloration. In the abdomen, you're going to find it, a couple of genes that are there in the abdomen. Now, if you look at the organisms on the right, you have a transcription factor which is going to cause gene expression in the wings now. So the area of the genetic code that codes for wings includes that color gene, so you get a wing spot there. In the abdomen remains the same. So you have a black abdominal band. So in this particular fly, you have black wing spots and a black abdominal band. Now, in another species of fly, you don't have that yellow gene being turned on in the wings. So the yellow remains off. But in this particular fly, the binding site for the gene has been lost in the area of the embryo that develops into the abdomen. And so in this case, you have a fly that has not only no wing spots, but no abdominal band. So when you move from the ancestral species that's on the left to the species that arise from it, one species has got wing spots because of an addition of a gene to a part of the body, and you have another insect that has a different coloration pattern because of a subtraction from a gene from a part of the genetic code. Okay, so comparing DNA and protein sequences, this gives you a lot of detail that even fossils can't give you. So this can determine evolutionary relationships. And if there's a conflict, if people perceive a conflict between genetic evidence and between morphology or the form of animals, genetics wins because it's simply more accurate. So the reason why is because if you have species that aren't related, how do you evolve precisely the same DNA and proteins sequences by chance? The answer to that are so small that you might as well call it impossible. So in our case, we have an additional amylase gene, and the reason why is because we eat more starch than chimpanzees do. We had more of a need for that. So when a person, when a human ancestor, just by random chance, had their amylase gene duplicate, that was an advantage because you could produce more of an enzyme that digests starch. It allows you to take better advantage of the resources around you, and so those individuals survive and pass on their genes. So you can take a look at homologous protein sequences. So cytochrome C is a protein that's found in mitochondria, and this is present in all eukaryotes. And it's a very important gene, or a very important protein. And if you mess with the sequence of that protein, most of the time you're going to get a really bad mutation from it. But over time, you can accumulate differences that don't affect the function of the protein. And so the more amino acid sequences between species, the more distant that you are from a common ancestor. So if you take a look at this, from chimpanzees, we've got no differences. From a rhesus monkey, we've got one difference. From a rabbit, 9. From a cow, 10. From a pigeon, 12. From a bullfrog, 18. From a fruit fly, 25. From yeast, 40. And when we went over that whole penny evolution thing, you can see that this follows the number of amino acid differences from humans follows right along with how distantly related that these organisms are from us. So DNA will accumulate random mutations at a regular rate if those mutations are not selected for or against. And this means that over time, you get more and more random mutations. So you can have a species that differs in several portions of its genetic code from the original species and from each other. So if you estimate that a particular gene mutates once every 25 million years, if you have two differences from an ancestor, this could indicate that those ancestors are approximately 50 million years apart. This is something that's called a molecular clock. And there are certain genes, especially in mitochondria, that tend to stay pretty stable, or genes that are involved in protein synthesis. So again, two species that had the same common ancestor 50 million years ago, they might have 4 differences in the nucleotide sequence of the gene.